Hello, my name is Michael Nunez. This is a pre-recorded talk for the 2021 Virtual Mathematical Psychology Conference. This research was conducted jointly with Ramesh Srinivasan and Joachim van der Kerkhove at the University of California, Irvine. I will be at the University of Amsterdam, where this research will continue. The title of my talk today is Recovering Parameters of Joint Models of Human EEG and Behavior During Decision-Making. In this talk, I review some of our recent work on understanding individual differences in decision-making cognition. In particular, we review statistical computational modeling of human EEG and behavior. So let's ask the question, what is the best method to understand individual differences in cognition? First, let's review our previous work. Computational models of human behavior during decision-making tasks have been developed in order to better understand human cognition. Human behavior in these models is encompassed by response times and accuracies obtained from simple experimental tasks where a participant must make one of two choices. For instance, in this task, the participant observes a grating image in visual noise and must then decide whether the grating is of high or low spatial frequency. That is, whether the parallel bars are close or farther away from each other based on previous training. The participant then makes a response by pressing one of two buttons, and then accuracy and response times are recorded. Now, the participant then makes many responses over multiple trials, and we collect response time distributions for both correct and error responses. These models are controlled by parameters that describe the cognitive process. For instance, there's a parameter that splits non-decision time and decision time. There's a boundary parameter that controls which choice is made after enough evidence is reached, and by changing this boundary results in the speed-accuracy trade-off. And then finally, there's a parameter that encodes the average rate of evidence accumulation for correct response, or the ability of a participant to collect evidence that encodes both the speed and accuracy of the participant. So confirmed in some experimental studies and simulation, these models can actually recover some of the information about the cognitive process from just behavior alone. Now, when we record EEG, we can get additional information about that cognitive process. For instance, from occipital electrodes, we can get participant average waveforms, the N200 latencies, which are negative potentials over occipital electrodes that we've shown reflect visual encoding time. And therefore, we can split non-decision time into visual encoding time and motor execution time. So this is one example of how joint computational modeling of both human EEG and behavior can further reveal human cognition. And in particular, individual differences in cognition by recording behavior and EEG from those participants. In this talk, we will focus in the cognitive process of the accumulation of evidence and best modeling practices for understanding individual differences in the accumulation of evidence. Possible EEG signals thought to reflect the accumulation of evidence include a widely studied potential over parietal electrodes called the central parietal positivity slope, the readiness potential, which is a motor-related potential over motor cortex electrodes. Influencing evidence accumulation is the visual attention, measured by steady-state visual evoked potentials using EEG, or SSVPs. In particular, signal enhancement and noise suppression are thought to be measured by SSVP amplitudes. So to better focus on one form of our question asked earlier, what is the best method to understand individual differences in evidence accumulation? So to answer this question, let's focus on two model classes. A directional model, which we had previously used in multiple studies, where the EEG influences cognitive parameters, which then in turn describe observed human behavior. To better understand the cognitive process of the evidence accumulation, we must need an integrative model in which model parameters describe both behavior and EEG. So now let's use integrative modeling to test neurocognitive theory. So why would we use integrative models of EEG and behavior to help differentiate individual differences in the evidence domain? Well, the evidence domain is typically thought to be made of stochastic samples of evidence, a continuous random walk process, typically estimated using infinitely small time step for convenience, which is called a diffusion or Wiener process. Each step of this evidence accumulation process will be drawn from a distribution with some mean given by the drift rate delta. The other parameters that describe behavior were previously mentioned, the boundary alpha and the non-decision time tau, which could be thought of, and often is thought of as split into two parts. However, we might suspect that individuals could have internal noise differences that influences individual differences in observed behavior. The noise of each sample is controlled by its variance, called the diffusion coefficient, given by this Greek letter variant of sigma. Typically, the diffusion coefficient is set to 1 because it cannot be estimated along with the drift rate delta and boundary alpha with just a behavior alone. We talk more about this in our 2015 and 2017 papers. However, by also observing EEG from these participants, for instance, the central parietal positivity slope and cortical desynchronization and synchronization as estimated by EEG power bands, where we summarize the EEG power bands with one summary measure over electrodes, we might be able to obtain more information about individual differences in the evidence domain that can actually be differentiated using all this data. So now one example model is the idealized model in which we have 3 EEG measures that correspond to 3 model parameters. Let's use hierarchical Bayesian modeling to understand and fit models to this data, where both RTs and accuracy distributions are described by a drift diffusion model process with parameters drift rate delta, boundary alpha, and non-decision time tau, and a mixture of a lapse process that we just assume it generates a 0 to 3 second response and also a random correct or incorrect response. And the amount of lapse process is controlled by the probability parameter theta. All 4 parameters are observed for each participant and condition, and RT and accuracies are observed for each trial participant and condition. We also observe CPP slopes, for instance, for each participant and condition, and we assume that these CPP slopes are generated by a linear mixture of drift rates for each participant and condition. We could also assume that an EEG power band is described by a linear combination of the boundary parameters. And then we can assume that the drift rate for each participant comes from a normal distribution with mean given by the condition just for a better estimate of these drift rates across participants. And we can do this for the boundary, non-decision time, and the lapse probability. However, if we also observe an additional EEG measure, we can estimate the diffusion coefficient well for each participant. So let's say we have an EEG power band, a third EEG power band that is described by a linear combination of diffusion coefficients. And again, let's assume that diffusion coefficients come from some hierarchical distribution. Okay, so now we have an idealized model. Now let's stress test this integrated model with simulations. Okay, so now let's take our idealized model and remove 2 of the data types. So now let's observe a model in which we only have one EEG power band that we assume is described by a linear combination of diffusion coefficients. Okay, so now let's simulate this model where we've picked mean boundary parameters, mean non-decision time parameters, mean drift rate parameters, and diffusion coefficient parameters for each participant and condition. We have 33 participants and 3 conditions that we are simulating from. And we also assume some linear relationship to EEG measures where we step through various correlations to these EEG measures. In simulation, we also allow the boundary, non-decision time, and the drift rate to have trial-to-trial variability. Okay, so now let's estimate this process by drawing samples from the diffusion process, where each sample from the diffusion process will give us an RT once that evidence path crosses a boundary. Let's do this multiple times. And then let's find our simulated RT distributions and simulated EEG measures where our simulated EEG measures are just one value per participant and condition. Okay, now let's take our simulated data, both EEG and RTs, and find parameter estimates. to this simulated data and see if those parameter estimates correspond to the real parameters. So we found parameter estimates for the boundary, parameter estimates for the drift rate, parameter estimates for non-decision time, parameter estimates for the diffusion coefficient. And we also have estimates of the linear relationship to the EEG. Now, when we look at the recovery of our generated diffusion coefficients for each participant and condition, where the x-axis here is the true diffusion coefficient, collapsed across participants and conditions, and the y-axis is the parameter estimate, in particular the posterior distributions of those diffusion coefficients. We can see that we can understand a little bit of the true individual differences in these diffusion coefficient parameters because we are tracking the orange line, which corresponds to a true recovery of these parameters. However, given by the dark blue and teal lines, which correspond to the 95 and 99% credible intervals respectively, we can see that there's quite a lot of uncertainty about those parameters when we fit the model to the data. Also of note is that the green star and the black circle are the mean and median respectively. We also find recovery of drift rate, where again the x-axis is the true drift rate and the y-axis is the parameter estimate. However, we do not recover the boundary parameter when we only have an EEG correlate of diffusion coefficient. So on the x-axis, we have the true boundary, and on the y-axis, we have the posterior distributions, and we can see we're very uncertain about the boundary, and we don't recover the true relationship. So while this model converges, we actually not getting any additional information that we would have just with behavior. And our lapse parameter is not recovered, although we're not so interested in that parameter. Okay, so this model may only be useful for EEG and behavior prediction and not for finding individual differences in cognition, in particular the evidence domain. Now, instead of an EEG measure being described by a linear relationship to diffusion coefficient, let's instead allow an EEG measure to be described by a linear combination of the boundary, and otherwise keeping the exact same model. Now we actually do get some recovery of the diffusion coefficient parameter. In particular, we can still somewhat estimate true individual differences in these diffusion coefficients across participant, even though the real diffusion coefficient parameters are not recovered. This is also true of the drift rate, and it's true of the boundary. It is not true of the lapse parameter. However, we're more interested in the evidence domain. So what this shows is that this model would be useful for finding individual differences in cognition in the evidence domain. And in particular, the diffusion coefficient model had a correlation of 1. This boundary model only has a correlation of 0.75 that we've generated. So we're stepping through multiple different types of correlations to see how well this model can recover in different scenarios of noise in the EEG and in the behavior. And we're finding that this model is somewhat robust to noise. So this is exciting because this means that integrative neurocognitive modeling may actually be helpful for better understanding relative individual differences in cognition. In the future, what we'll be doing is stepping through different correlations of EEG to model parameters, as well as injecting noise into the behavioral model itself in order to figure out when EEG is actually useful in combination with behavior in neurocognitive modeling to understand these individual differences. So thank you so much for listening to this talk. I thank my collaborators on previous work and current work, Kitty, Mariel, Steve, Ashwarya, and Jessica, of course, as well as Joachim and Ramesh, who are co-authors on this work. More information about our previous studies can be found at my website /publications. Directional modeling in JAGS and STAN, we have some examples on GitHub. This work was supported by the U.S. National Science Foundation. Thank you so much.